Hello, Bio 131 people. This is the third time I've tried to record this video, so here's hoping the third time's a charm. So, for the next week, we are going to be going to, let's see, go to the coronavirus assignments for spring 2020, the week of 3:30. So this coming week, and then the following few weeks, we are going to be basically composing a scientific paper over your Daphnia experiment section by section. So each week is going to be a different section. So this week's focus is going to be on the introduction. So I've uploaded a couple different documents here. So you have your Daphnia experiment because I know it was a long time ago since we had, viable, cute Daphnia that we could look at, check out their heartbeat. So I would definitely encourage you to reacquaint yourself with that once it loads. Here we go. Hopefully this looks familiar. So our purpose is really just looking at how different chemicals affected the heart rate of the Daphnia. So read through that again, and it can give you a lot of good ideas for things you might want to refer to in the introduction, which is what this week is going to focus on. So the other item that has been uploaded is a guide to writing a scientific paper. So you guys are going to be doing a lot of this as you have like bio majors, different classes. You'll probably even get this handout again in Bio 105, maybe even in Bio 106. It's an extremely handy document, and it basically takes you through every single thing you could possibly need when composing a scientific paper over an experiment you carried out in a lab. So if you want to go ahead and save this somewhere or print it out just so you have it, you'll definitely need it again. It will definitely serve you well. Fun fact, this actually was the same document I used whenever I was starting off at SIU in the basic biology for science majors in 2002. So very little has changed in terms of the elements of a scientific paper between now and then. All right, so this takes you through the different sections of a report, and then it also is going to give you an example of a study. So our example here is looking at the effects of hair dye color on mutagenicity of Salmonella typhimurium bacteria. So right up my alley. All right, so you have a very good example of the different elements that are included in an introduction, which is what you're going to do this week. So basically, with your introduction, you are telling the reader basic information. So why would anyone care about this to begin with? How is it relevant to biology? What are some potential applications? Any type of background information that you may have read that led you to your hypothesis. So the hypothesis generally is going to be stated in your introduction, and you're probably going to have a little brief overview, not a detailed like step-by-step overview of the experiment, but just kind of a glossing over of what you did to study your particular hypothesis. And then at the end of that, usually you're kind of referencing your hypothesis. So if the hypothesis is correct, then we would expect to see this at the end of our study. So this is an excellent example. So it tells you, a little bit about, what you're going to need, and then you have their example. So here with the different elements, for this experiment, they stated their hypothesis. They also explain a little bit of the background information that led them to generate their hypothesis. So your literature review, source sleuthing skills will be helpful here in finding sources that you can use in your introduction. Generally, you will have cited sources inside of an introduction. Typically, they're going to be primary sources because these lab reports are kind of the basics of a scientific journal article. So you're also going to include, the background that would help someone understand the why. So why would we care from a biological standpoint about any of this? For us with the Daphnia, usually when you're doing some type of scientific research and you are looking at chemicals, you're probably going to want to use a simpler life form first before you just start, bombarding a person with epinephrine and caffeine and sucrose, just, to see what happens. Usually you'll have a smaller, simpler animal model that you will be working from. So you will have a better idea of what's going to happen with a human trial, for things like that. So once you've gone through that, you'll see down here that they have given a very brief summary of kind of what they did, how they approached their hypothesis, how they went about studying it. And so usually they will have a prediction. So if our hypothesis is supported by our experiment, we would expect to see such and such as a result. And that is basically an introduction. So it's just kind of a nice overview to the relevant, uh, any background information that led you to the hypothesis that you're testing, and then a very brief, kind of synopsis of how you're going to be testing your particular experiment, which for us is our Daphnia. All right, so then if you look further, they have a lot more. They've got figures in their introduction. I mean, that's absolutely fine if you wanted to include a figure of maybe like Daphnia anatomy, because remember their hearts are in a weird location from ours. They're on their back. So that would be, something that you definitely could include as one of your figures if you wanted to. All right, so here's where they've gotten to their predictions. And then once you make it to that point, really the next section is going to be your materials and methods, which we will get to the following week. So with your introduction, you likely are going to have some references. I'm not going to require you to turn in a references page with the introduction. However, I do want you to have the in-text citations in APA format for the different sources that you're going to use. And I'll get the sources when you guys turn in the entire complete paper a few weeks down the road. So if you look at this one, you can see how they have like a Greenville News that was cited. They also mentioned the textbook. So the Campbell Reese biology, which is what you guys will use in Bio 105, was mentioned here. And you've also got a couple of other references. So if you use any outside information, and typically you want them to be not just please no Wikipedia, use all of the skills that you learned from Jamie when she was there to talk to us about library resources. If you wanted to use a textbook, I mean, just because we're doing a very simple paper, you can do that, but please try and have at least 2 primary sources. That's what I'm going to require, 2 primary sources somewhere in your paper. All right, so also included in this lovely guide to scientific writing, if you scroll through the different sections of the paper, you make it to a page that has a checklist. So this checklist is sort of awesome. As long as at the end of your scientific paper, you can kind of check off each of these boxes, you're probably going to do fairly well in terms of your grade on your scientific paper. So since we are just focusing right now on the introduction, things you want to be sure you can check off. We want to be able we can state our hypothesis. We're gonna want to give, some solid reasoning for why we have the hypothesis that we do. We are going to be explaining biological relevance, the biological basis of this experiment. You are going to have in-text citations, and then you are going to have kind of like a brief overview of how you are going to test your hypothesis. And then finally, so if your hypothesis is supported by this experiment that you are carrying out, then what do you expect your results would be? So those are the different elements of the introduction. So the next week will be materials and methods. I have a feeling that you guys turned your data in to me so that I could keep it for you since we were going to have spring break before we started our data analysis. So if that is the case, someone please let me know. I am going to be going back to my office a couple times next week. So if I have your Daphnia data, please tell me and I will find it so that I can, I don't know, scan it to you or get it to you in some form or fashion so that you can do the entire paper. All right, and as always, if you have any questions, anything at all, please let me know. Use the discussion board, that way everybody can see it. And good luck, guys, and stay healthy.